Yeah, that's great.
Okay.
Good morning, everybody. Thanks for bearing with us. Thanks, Maria. All right, I'm going to get my screen started. We have a lot to cover today, so I'm going to quickly go right over to that right now so that we can get started. Hold on. All right. Share screen. Beautiful. All right. Welcome everyone to our summer installment of the Knowledge Portal Network webinar and workshop. I'm going to get my full screen up in just a second here, and I'm going to get going. All right, I'm hoping folks can see me. Please place in the chat if you can't. I want to make sure everything's functionally going well. Beautiful. Thank you, Edgar, for telling you can hear us. That was a big help. All right, folks. So today we have three amazing speakers I'm so happy to have. We are featuring our lung genetics knowledge portal and the Lung Epigenome resource today in our webinar. A slight departure for the AMP-CMD activity, but this is really important because this is the expansion of our platform for human genetics and genomics into other common diseases. And I think this resource is valuable to sort of all of the community of complex trait genetics. So first we'll hear from Michael Cho today about the impetus and the reason, sort of the background of lung disease genetics. And then Maria will demo the actual portal and how you can use some of the data that Mike will talk about and how the portal will hopefully advance to help navigate our way through lung disease and the regions that have been identified from genome-wide association studies toward function. And then Kyle, we're very lucky Kyle Gaulton will speak as well about the resource he built for the Lung Epigenome Atlas, which is a similar one if those of you who are familiar with the type DGA or the Diabetes Epigenome Atlas. It's built on the same platform, but specific for lung disease. And this is a great thing because this will fully integrate with our knowledge portal and make those results accessible to you. In the end, we'll talk about what's to come. But briefly, I'll quickly start with my typical news from the network, what's broadly going on in the Knowledge Portal Network and broadly in AMP-CMD. So a couple of exciting things. As usual, if you need to find any resources and tools, they're all on our resources page. Our next webinar is going to be September 23rd when summer is over. But if you need any resources and videos and tutorials in the meantime, you can find them on this page. We also have added contributor videos now, which are done by the scientists themselves to talk a little bit about the resource, the science, and how you can use it on the portal. Other resource development, you're building portals in other areas of biology. We think this is really valuable as we move out of human genetics. We've been very lucky to partner with Toby Walter and Bob Faris to build a portal for an organelle, specifically related to lipid disease and cardiovascular disease. The Lipid Droplet knowledge portal just went live a couple weeks ago. You can find a bioRxiv paper right there below if you're interested in reading more about it. This is an open access resource, but it really sort of starts to spread our wings into the transcriptomic, proteomic, and imaging world of biology to help represent results from those types of data sets. Moreover, for genetics speaking, we're expanding beyond the typical genetic models to non-added effects, working with CHEMA to represent results for complex traits, diseases in non-added effects. This is a slightly different departure from how we currently represent genomic associations on the portal, genetic associations. So we're using it as our research area to build its own portal to represent this and how you would navigate that, ultimately connecting with the broader resource. Another one is a brand new area for us. As you know, we've been involved in an ALS portal for several years now, and working with our community, we've noted that while there's a lot of work in the neurodegenerative space to do genetic association studies and functional genomics in the various diseases, there is a limited in resources that bring all the neurodegenerative phenotypes together under one umbrella and make them accessible. So we have a sort of a burgeoning effort in that area, working with folks in the neurodegenerative space to help represent Parkinson's disease, Alzheimer's disease, frontal temporal dementia, and ALS in a knowledge portal. So look for that coming in the fall. And then finally, out this month, last month actually, is effector predictions, working with cardiogram for coronary artery disease. This was its own portal, but we've encapsulated in the CMDKP for you to peruse those results, and the paper is there if you're interested. We'll make these slides publicly available on the portal shortly after this webinar. So on the horizon for our mothership, for AMP-CMD, as some exciting things really, and this will expand across all the platforms, are new ways to visualize integrated genetic and genomic data. This is something that's been on our radar for a while, but we really are making some nice tangible progress in that space now with the extension of our GEM visualization to bring together genomic annotations, genetic associations, credible sets, and the various bioinformatic method results that allow you to navigate and prioritize genes and variants. And this is just a quick teaser of the mock-up that DK on our team is working on to help you as a user, if you're interested in genomic region, to prioritize variants within a given region based on credible sets, various annotations and enrichments, and developing for you a means to navigate that in hopefully a user-friendly way and with access to the underlying data and results. This is just a mock-up. In September, we hope to actually share with you our first vision of that for going forward with ANT-CMD. So with that, I'm going to move on and turn this over to Michael, who's going to talk about the lung disease knowledge portal and the epigenome resource, and then Maria and Kyle will follow on. So Michael, welcome. I want to thank Michael. Michael actually started working with us about 2 years ago. He came to us with this idea to expand, and it's been really exciting to work with him to understand what is needed in that field as a gap to try and make results accessible for lung disease and other traits. So thank you, Michael.
Thanks, Noah. I'm really appreciative, and again, admire your work so much, and one of the reasons I obviously came up and tried to find you guys. So I've been given the task of surveying, I think, respiratory genetics quickly, so there's obviously a lot I'm going to leave out, so I apologize. I would actually start to thank that there's a lot of people, and we've created somewhat of an advisory team or board here with Benji Raby and many other people really around the world and from different disease disciplines, including people from the NIH who have also been funding basically all of our work. And then of course the Knowledge Portal team. So why do we care about respiratory disease? You know, we're not cardiovascular disease, but we're actually honestly pretty close. This is from the WHO, where chronic obstructive pulmonary disease has now risen to the 3rd leading cause of death globally. Lower respiratory infections are number 4. So there's a huge burden of disease in the lung. And if you break this out, this is now from just Europe, you can see that there's a different array of diseases here, from COPD and bronchiectasis, asthma, lung cancers, et cetera, et cetera. And so myself as a pulmonologist, and we as the lung community, I think have an enormous task ahead of us. I wanted to scan through a couple of the respiratory diseases that we consider. So chronic obstructive pulmonary disease is what I study. This is, it has some similarities to asthma. The difference is it's really irreversible inability to exhale. And people usually associate with smoking, and it's seen in conjunction with emphysema. So when people talk about emphysema, they often also talk about just COPD generally. I think asthma, many of us are familiar with. It's prevalent in childhood, but also adulthood, and people often associate asthma with other environmental triggers like allergens. Interstitial lung disease, the most common one of these is called pulmonary fibrosis, is not something that most people think about. So it's because it's pretty rare, but the mortality of pulmonary fibrosis is actually higher than most cancers. Many of us are familiar with pneumonia, but we should know that pneumonia is a heterogeneous condition. It can be caused by bacterial causes, viral things such as COVID-19, and then ARDS is a it was an uncommon manifestation, but a very severe manifestation of pneumonia, and so there's overlap between all these infectious pneumonias. Lung cancer is actually the leading cause of cancer mortality, despite what we hear about breast and prostate cancer. Lung cancer is actually the biggest killer, and increasingly something we see in non-smokers. We have another disease here, pulmonary vascular disease, pulmonary hypertension. This is a rare disease, but it affects young people and can basically cause their heart to fail and require heart and lung transplant. Bronchiectasis is a condition with chronic inflammation, infection, and dilation of the airways. One of the manifestations of cystic fibrosis and pulmonary ciliary dyskinesia, which we think about as genetic conditions, is bronchiectasis. And we can also see other conditions leading to bronchiectasis, and that's just sort of, some of the top ones that I was thinking of, but there's many others, including critical illness. There's overlap between sleep disorders and lung disorders and control of breathing, et cetera. So what about the genetics of these? There have been genetics of these diseases at least. So COPD, I've been studying. So alpha-1 antitrypsin deficiency is the prototype genetic cause of COPD, known for over 50 years. But you also see COPD and emphysema and other Mendelian disorders. And we'll talk a little bit more, but there's common variants and a lot of overlap with lung function based on how we define COPD. Asthma, I think, is a prototype for many. There's a Nature paper, which was one of the first demonstrations of an EQTL in ORMDL3 with asthma. It's really on classification, even though it's a lung disease, there's a lot of overlap with autoimmune disease, and there's differences in childhood and adult. Pulmonary fibrosis is known because of telomere pathway rare variants, as well as this really outsized odds ratio MUC5B variant, which is common, has an odds ratio about 5. When we start talking about pneumonia, there's actually smaller sample sizes, even though this is a common condition, just because of the fact that this is a episodic thing. People have done specific GWASs for tuberculosis and COVID. Lung cancer, there's been a lot of great work, Chris Amos, Christiani, and others, which actually have some shared loci with COPD and telomere pathway genes. And there's been some smaller studies of pulmonary vascular disease, but really not for bronchiectasis, some for genetic modifiers of cystic fibrosis and ARDS and others. But as a non-respiratory investigator, why would you care about respiratory disease? So I've already told you about how it's a leading cause of mortality. It's also a rising cause. We don't have any new treatments for COPD that have been developed that actually can attenuate mortality. There's no drug right now that pulmonologists agree upon will reduce the progression of decline of lung function or mortality. They're a major source of health disparities. Some of this comes from the fact that occupational exposures and pollution and things like that affect lung conditions. But if you look at things that are predominantly affecting lower income people or people of underrepresented minorities, lung conditions are at the top. I already mentioned environmental exposure, so it's a rich area for looking at things like gene-by-environment interactions. I would argue, because I'm a lung investigator, that a lot of the lung diseases are relatively understudied for their burden. And I would also say it's a challenge. The lung has a huge array of different cell types. There's issues with environmental exposure, lung development, and things like that. I think it's actually a very challenging thing to study. Unlike, I would say for diabetes, for example, there's a lot of focus on the islet cell. We don't have a similar cell that we know is the pathogenic cell in COPD. So I study mostly COPD. I'm going to talk about it a little bit more in detail. So this is dyspnea, airflow limitation, often exposures to noxious stimuli. And here's a picture of a COPD lung with these air sacs that become dilated and plugged. Has a large burden. And if you didn't know anything about COPD, know that Spock actually died of severe COPD. Most people think of it as smoking, and you see pictures like this saying it's all due to smoking. But if you take people with similar cigarette smoking exposure, this is a measure of lung function. There's often very different lung function, even with people with similar pack exposure. So the rationale for studying the genetics of COPD is it's increasing despite tobacco control. It progresses even when you quit smoking. There's no specific therapies, and the reasons that there's varying susceptibility are poorly understood. The first thing I think a lot of geneticists will think about is what is the heritability? So here is actually a sibling relative risk from a Boston early onset COPD study saying that you're around 4 times as likely to develop COPD if you have a first degree relative with severe COPD. This is a twin study from Denmark and Sweden showing about 60% from twin studies. And then we have from genome-wide association study data around 40%. So there is a significant portion of heritability from COPD. We've done our usual thing where we've accumulated more and more sample size through collaborations with a lot of people and gotten from just a handful of loci through the International COPD Genetics Consortium and UK Biobank somewhere up here. This is our latest GWAS led by Huna, Dimitri, and Brian, and this is our Manhattan plot. I will make an important point here, which is the way we define COPD for these studies and others is actually by using lung function. And so at the upper left, you see a spirometry curve, which is what happens when you take a tube and you blow into it and have somebody measure that. It's probably not surprising to some of you, but it was surprising to a lot of people in the field that if you take a population and just look at what defines lung function in the population, including in non-smokers, that a lot of those loci actually overlap with those for COPD. And studies led by Nick Shrine, pictured here on the right, Louise Wayne, Martin Tobin and colleagues, as well as Stephanie London's group from Charge and others, have done large studies looking at this quantitative trait. And if you're just looking at a quantitative trait, you're going to have a lot of power. Their latest paper, 279 signals in a latest European ancestry analysis, and most of these are also informative for COPD. You can actually see the correlations with FEV1, FVC, and FEV1, which are used to define COPD with each other, as well as the correlation with FVC, which is another lung function measure, and peak expiratory flow here. So a lot of the lung function measures are correlated. A lot of them are informative for COPD. I'll talk a little bit about asthma. I think asthma genetics is maybe one of the oldest for common respiratory diseases. It's very common. It may not have the same mortality impact, but it has a huge burden of disease, over 300 million people worldwide. We've actually thought about it as prevalent mostly in Western countries, but it's increasing in these other countries as well. As I mentioned, there's disparities between different races and ethnicities, and also interesting differences between men and women. And we can think of asthma as things occurring intrauterine, also in development, and also things that affect exacerbations, and genetics are probably important for all of these things. The discoveries for asthma recently reviewed in this Lancet Respiratory Medicine paper, actually again, similar with increasing sample size and good phenotype definitions, you're increasing your discoveries. A lot of these fit into the known paradigms for asthma and immune disease and existing or new drug targets. Some of these include TSLP, IL-7R, and CRLF2, which are actually currently in clinical trials. So this is the great example that we have of genetics being a good support for clinical trials. There's also really interesting things like this IL-33 rare variant that leads to lower asthma risk and doesn't seem to have any adverse effects. So again, an important sort of PCSK9 similar example. But as for most diseases, the actual target for most of these loci is not known. This is a diagram where the authors of this paper actually try to deconvolute some of this asthma, assign a causal gene, and identify some drug targets. Pulmonary fibrosis is a really interesting genetic disease, and as I mentioned, has a really high mortality. It's called a diffuse parenchymal lung disease, so it basically affects the entire lung and the lung tissue itself instead of just the airways. The way I typically think of pulmonary fibrosis is idiopathic, as opposed to things like asbestos, which can cause a similar pathology and phenotype. There's actually a whole family of interstitial lung diseases which have these really challenging to remember names, but they do share a lot of things in common. And we think of them often, as pulmonologists, we think of them in conjunction with COPD because they both increase with aging, and cigarette smoking is a risk factor for both. But the pathology is very different. This is a CT scan. It's a normal CT scan. So this is a cross section. You can see the right lung here, the left lung, and the heart in the middle. And this is what a normal lung is supposed to look like on the left. Oops, sorry. In the middle, you actually see a CT scan of somebody with severe COPD. You see there's basically, this black means there's basically no lung here, no lung here, no lung here. And the little lung that's present has big holes in it. The one on the right is actually a CT scan from a fibrosis lung, and you can see that there's all this extra white, and this is actually the tissue becoming scar and fibrosis. So the pathophysiology is actually the opposite, and it's actually much rarer. But pulmonary fibrosis has really interesting genetic signals. And as I mentioned, this MUC5B with a allele frequency of 10% has this enormous odds ratio and gives you a p-value that is not even displayable on this Manhattan plot from Richard Allen and colleagues. This is the largest GWAS of pulmonary fibrosis. You see RTEL1 here, which is, and TERK, which are some of the telomere pathway genes, as well as a lot of other really interesting signals. The other thing that's kind of notable is that you see FAM13A and DSP here, and I don't expect you to remember, but these were also present on the COPD plots. So it turns out this is a locus zoom plot of FAM13A from COPD. This is a locus zoom of FAM13A from a 2013 paper of pulmonary fibrosis. This is the DSP locus from our COPD paper. This is the same one from pulmonary fibrosis. So there's some relationship and then some overlap here. If you do a systemic evaluation, this is from our 2019 paper, and you use GWAS-PW, you actually find 5 regions that overlap between COPD and pulmonary fibrosis. And on the right here are several loci that overlap between COPD and asthma. We've also looked at other phenotypes here. And this is from Nick's paper showing that if you do sort of a FEWAS across UK Biobank, you're finding a lot of overlapping phenotypes. Interestingly enough, COVID-19 host genetics, hopefully many of you are part of this initiative. It's really wonderful. And the transparency, I think, is great. I mean, they're basically just, they keep opening up their results. So they're publicly available. CANZEL1, this one locus actually overlaps with both COPD and fibrosis, or all three, lung function as well, and is a risk factor for COVID. DPP9 is a risk factor for pulmonary fibrosis and overlaps, and there's other suggestive, at least, associations with MUC5B and surfactant protein D. So again, you have these intersections of these different lung diseases, which is really, really amazing. I think as many of you know, each of these associations is an opportunity for discovery because we don't know what's going on with most of these genes. This example from HIP, this first locus that we discovered for COPD, and also with lung function, we ended up doing chromosome capture to show that the SNP region interacted with the HIP promoter. We later found novel effects of HIP that were not necessarily related to the hedgehog pathway. And while it's cigarette smoke-induced emphysema that we look at in the mouse model, it's also age-induced emphysema, suggesting this is not just a developmental gene, despite the fact that it's hedgehog. We obviously need to do this for all the other loci. This is our approach that we did for our 2019 paper, looking at all these different kinds of data. But this was a sort of one-off sort of bespoke analysis for what we did. And we also looked at different phenotypes, again, just for that association. So I hope I've just shown you that respiratory diseases have a large burden and unmet therapeutic need. And if you're not a lung person, we welcome you to the community. A lot of the people who study lung disease right now were former cardiovascular disease investigators. There's a lot to do. I showed you some genetic risk factors for COPD, lung function, asthma, interstitial lung disease, and COVID-19. There's others. We are working on larger and more diverse association studies. There's a lot of related phenotypes that I think we can look at, including imaging and exacerbations. And I think that, as I mentioned, there's so many different lung cell types trying to get functional genomics of those, or getting even chromatin profiling and things like that is really important in what we need to do. So, you know, we had these GWASs, and one of the initial thoughts that we had is, oh my God, everyone's going to be studying these GWAS loci. And that didn't really happen. And people were really interested in their genes of interest. In fact, I recently had a grant that hopefully will be funded on a pathway related to hepatocyte growth factor. And, you know, using the portal, we were able to actually look up specific genes and find some significant and some sub-significant hits, but enough that it was actually pretty convincing that this is a pathway of interest. And so how do we connect?
Michael, I'm sorry to break in, but it seems that your slides are not advancing. I'm seeing the COVID-19 slide.
Oh, interesting. That's weird. Okay, hold on a second. Let me do new share. Did it advance before then?
Yes, they did.
Okay. Can you see this last slide now?
It just says you started screen sharing.
Okay.
You did a great job of describing, so I'm going to keep going.
Okay, yeah, I'm just going to finish up. It's just, this is the last slide. But I think there's a lot of, we really want to encourage other investigators, both basic science, translational, and even omic investigators, to be able to look up their locus of interest. We want to be able to view the overlap between the different lung phenotypes, and we want to facilitate the application of new genetic data analysis tools. And I think that this is where the portal can have a really, really big impact. So thank you.
Beautiful. Thank you, Michael. That was awesome. Maria is now going to walk you through what we've built so far with Michael and Benji and other folks in the lung community's feedback to try and get at, you know, the gap, which is making genetic data accessible and layering with genomic annotations so that you can prioritize genes and variants and visualize these in a way that hopefully a non-expert in genetics or in lung disease can navigate. I can see your screen, Maria, so that's awesome.
Great. Okay.
Beautiful. Take it over.
Okay. All right. So let me just check the time. I'm going to leave. Be sure to leave time for Kyle. Yeah, so this is the Lung Disease Knowledge Portal, and you can access it at lungdiseasegenetics.org. And if you know our other portals, it's very similar to that in organization. So if you know how to navigate the Common Metabolic Diseases Knowledge Portal, this is much the same. So let me start out by showing what genetic association data we have in the portal. So we add genetic association data to portals by phenotype or phenotype group. And right now we've only added phenotypes that are under the lung category and the COVID-19 category. But I want to stress that we can easily add other genetic associations from other phenotype groups to this portal. So we would really appreciate hearing from your community, what would be other phenotypes of interest that you would like to see in this portal with the other phenotypes. So right now we have the COVID-19 host genetics GWAS, but I know that a new release of these analyses has just come out, and so we're planning to update and add those instead. And we also have some asthma meta-analysis. We have lung function, COPD, and IPF. So that's what we have at the moment. And for epigenomic data, we have quite a bit of epigenomic data. We don't have a page listing that as yet, but we will in the future, and particularly when we start to load data from the Lung Epigenome Atlas, which you'll hear about in a couple minutes. And we're actively working on getting that into this portal. So I just want to quickly show you one of the core pages of the portal, if you don't know, if you're not familiar with these portals. So let's look at DPP9, which is an example that Michael mentioned. So if you type a gene name into that search box on the home page, you get to a page that centers on the gene and shows also 50 kilobases in on each side of it. You can look at a larger region very easily by clicking this button. But at the top of this page, you will see the strongest genetic associations in this region. All of these genetic associations are meta-analyzed in our software system. So we take every data set that has an association for a particular phenotype and meta-analyze them together using and it's an overlap-aware meta-analysis. So if the sample populations in different data sets overlap, this takes that into account. So the p-values that you see here we call them bottom line p-values. Now, in this display, we're also clumping associations by linkage disequilibrium. So this is a way of simplifying this display. It considers all genetically linked associations together and just displays the information for the lead variant, the most significantly associated variant in that clump. If you want to see the individual variants that went into the clump, you can click this box and see them, and that's quite an extensive list. If you scroll a little bit further down the page, here you will see the individual variants that are associated with this phenotype. And the phenotype you'll see by default is the one for which there is the most significant association in this region. But you can look at more phenotypes or change phenotypes. In this way, you can just start typing and it will filter the phenotypes based on what you have entered, and so you can add another phenotype to this display, to this table, and see the associations, and you can do that for multiple phenotypes. Then below this is an interface that is really an important one that we are continually developing, and we were interested in your feedback on how to develop it better, but we hope that this will become kind of the center of these portals for exploring the genetic association data in concert with all the epigenomic data. So when you add phenotypes up here, you add locus zoom plots here. So you will see the plots for of associations for all of these phenotypes. And you can also look at epigenomic annotations using this plot. So I'm going to get rid of this plot just to make this display a little easier to see. So if you want to see epigenomic annotations, you can add all annotations from a tissue group. And these are kind of broad tissue groups that have more specific tissues within them. But let's look at lung. And then you have added, okay, you have added tracks that display the regions that have epigenomic annotations. And the best way to look at this is to split the tracks. And this will then show you a separate track for each of these regions that are predicted to be promoters, enhancers, accessible chromatin, or transcription factor binding sites. And when you mouse over the plot, you get these nice crosshairs. So you can see, for example, here that the index variant in DPP9 overlaps with binding sites and predicted promoter, and looks like probably accessible chromatin as well. You can simplify this display by increasing the enrichment of the annotations. So all of these epigenomic annotations are subjected to the Gregor algorithm, which looks at enrichment of genetic association signals within these regions and in particular tissues. So we just added the criterion that the annotations must be at least twofold enriched. And here, now we can see that, that this promoter annotation, we still see it. So this is a stronger one. Another way to look at these is that if you are interested in just a particular type of annotation over any tissue, you can add those annotations. And again, the best way to look at this is to split the tracks. And here you see a separate track for each tissue group that we have. And again, you can line up, my screen's a little small here, but you can line up with the crosshairs and see what's going on in any particular region. Another way to look at epigenomic annotations and annotations in general is to go to the variant page. We have a page for each variant, and I've already queued up the page for this variant. So this was the index variant that we were looking at, and we have a table that has all of its annotations and predictions, the predictions of its impact done by the ensemble variant effect predictor that we run in-house. Also a PheWAS plot. So this is associations of this variant across all the phenotypes in the portal, and you can mouse over them and see what the phenotypes are and what the significance of the association is. Table of transcription factor binding sites that are predicted to be altered by this variant. And then finally, the annotated regions that overlap this variant. And again, you can filter this by tissue. So let's look at lung. You can see that this variant overlaps all three of these types of annotation in lung tissue. So there's a lot more to the portal, but I think I'm going to cut it short to make sure Kyle has enough time to talk. I'll just mention briefly that we do have phenotype pages. If you click on the name of a phenotype anywhere in the portal, let's find a phenotype name. You can find a link to the page. You can also search for this on the home page. And here you can see all of the meta-analyzed associations for that phenotype across the portal, the top signals, and so forth. Likewise, there's a page for each gene, and each gene has gene level association results across all phenotypes done by the MAGMA algorithm. So you can see if a gene is significantly associated with any traits of interest. Okay, and first I'll ask if there are any questions about this. Okay, you're welcome to ask questions later, or of course, email us anytime, or we have a contact form on the website. Here, I'll show you that too. Contact, this leads you to a form where you can put your question, and we're really happy to get comments and feedback and questions.
As we flip to Kyle, Kyle's going to demonstrate sort of a complementary resource, which has specific data for lung disease for epigenomic annotations. And what's lovely about this is that it's its own resource. We can access those results directly But you can also eventually, hopefully, in the coming months on the lung disease portal that Maria just showed, you will see the integrated annotations that Kyle is going to speak about here on the genomic region minor gem that Maria showed, such that you can navigate between the genetics and the genomics more readily. And there'll be links to this resource that we hope to expand with more specific lung disease data sets from the community. So that's where we're going to need the community to give us advice about what are the right tissues, cell types, and annotations to represent more specifically, and how to layer those with the right traits and phenotypes on the portal. So with that, we'll turn over to Kyle. Kyle, we can see your slides perfect, so that's great.
Okay, great. Thanks, Noel. Can you hear me? Good. Okay, so I'll give a brief overview of our lung genome browser that Noel mentioned, the goal of which is to annotate non-coding variants for complex lung disease and also to provide data to the lung knowledge portal to annotate variants within that resource. So I'll go relatively quickly, maybe have like 10 slides, maybe 10 minutes. So the key question that we're trying to address with this resource is that most common disease risk variants are non-coding and therefore have no immediately obvious mechanism. All the diseases that Michael mentioned, say maybe like rare Mendelian diseases, but the common diseases that Michael mentioned in his talk, the large majority of variants associated with those diseases are non-coding. And so understanding their function and what genes they're affecting, what cell types they're affecting those genes in, things like that are critical in order to understand the biology underlying these loci. Doing that requires effective annotation of the genome and the epigenome. So that was the goal and the sort of reason why we set up this resource, the Lung Genome Browser, and the website is lungepigenome.org. It's a data repository for epigenomic data, visualization of lung genomics data. It's based on open source software developed by ENCODE. It's also very similar to the diabetes epigenome site that we had set up, and I'm sure most, if not all of you on this call are familiar with that. It's just the same infrastructure that we're using for this site. And currently the database has data from one study, which is a single cell study of chromatin and gene expression in human lung samples, and it was Wang et al. In eLife. Now, I'm not myself an expert at all in lung respiratory genetics or lung biology. I work closely with neighbors, primarily Chin's son, who is a lung biologist, and then also Sebastian and Alan, who are in the Center for Epigenomics at UCSD. And so together we kind of set up this resource. And all of this effort in creating the resource and populating with data and making many of the tools that I'll quickly describe was done by Parl, who is also the primary person working on the DGA diabetes epigenome site. Okay, so yeah, so I'll just say, so we have, like I said, currently data from one single cell study. The goal over time is to increase the amount of data that's in the site through which we can give people the ability to view those data and also pass it on to the lung knowledge portal. So I think one key thing is, since I'm not overly familiar with the field, data sets that exist and studies that may be relevant for annotating respiratory disease risk variants, it would be helpful, to communicate with people who are maybe interested in contributing data or have publications that they think are valuable for this purpose, so forth. Please feel free to reach out to us, and the hope is that we grow this resource over time to be more inclusive of data across the community and help provide a more comprehensive annotation for annotating lung and respiratory disease risk variants. Okay, so I'll just give a couple slides about the data in this paper that is currently the data set that's in the lung genome site. So this is single cell profiles of accessible chromatin from human lung samples. In total, we have over 90,000 nuclei profiled of ATAC-seq. And here the cell types we identified, so a representation of the major cell types of the lung, as well as some subtypes within the cell types. And these cell types and subtypes are annotated based on their genomic profiles. Similarly, we have RNA-seq data from the same samples, fewer nuclei, but a greater ability to resolve cell types. So we have many additional cell types here beyond what we identified in the ATAC-seq data, and again, representation of lung cell types and subtypes. So these data give the ability to annotate regulatory programs within lung cell types and also the expression of genes within lung cell types, through which you can use these data to annotate risk variants for respiratory disease. Another piece of information that we derive that we get from ATAC-seq data is the ability to predict target genes of distal regulatory sites using something called coaccessibility. I won't go into the details here, but they're predicted links between regulatory sites and genes that we can drive within each cell type as well. So taking this information, going from risk variants that are non-coding that we may have little information about their function, we can start to layer these pieces of information. Again, in this study, accessible chromatin in individual cell types, coaccessibility linking those sites to genes. We also have predictions of variant effects on the activity of each cell type, which I didn't describe, and then the expression of genes in each cell type. You can start to layer these pieces of information to gain some insight into how non-coding variants may function in lung cell types. And so as just an example of this, one of the first, well, maybe the first actually, locus that was associated with, I think, severe risk of COVID-19 variants near gene SLC6A20. So we took the fine mapping data for this signal from the International COVID Consortium, intersected it with chromatin data from our study across all these cell types. And I think one of the risk variants overlap sites active in alveolar cells that was also linked in alveolar cells to SLC6A20. The expression of SLC6A20 was preferentially in alveolar cells. And so we can, using these data, can kind of hypothesize about how these variants may be functioning, and in this case, influencing risk of COVID. Okay, so I'll just walk through a couple of the tools that we've set up on this site to be able to interact with these data. Here's a list here of some of the things that can be done. So you can query and access the process data files if you just want to download them and use them yourself. View accessible chromatin profiles in a genome browser. View these profiles in what's called single cell browser, where you can view the embeddings for individual cells. View the cell type expression patterns of genes. View the links between distal sites and target genes, and also query genetic variants for overlap with all these annotations. So this is in addition to all the tools that you can use from within the knowledge portal as all this data is being aggregated by them as well. This may, in some cases, give you the ability to interact with these data at a slightly different level or in slightly different ways that is complementary to what the knowledge portal can provide. So this is just a view of the data. Like I said, this is just data at the moment from one study. So these are the sites from the cell types in this study, but you can view and download those. Here's an image of the single cell browser for gene expression data, where you can view the different cell types. You can select different cell types. You can view expression of genes across different cell types. And this is currently using the UCSC browser. We also have one set up for the accessible chromatin data. We're in the process of migrating this over to a browser that we think is a bit more elegant, that also has some additional features and tools, which is the CellXGene, which I believe is what's used more heavily by like CZI and Cell Atlas and these single cell based consortia. And so Paul's in the process of migrating the single cell browser over to CellXGene, which should further improve the ability to interact with the data in this way. Again, look for the expression of particular genes, compare the expression of selected genes across cell types, look at differential expression. So it has actually a fair number of tools from within CellXGene that you can use to ask questions of these data. And I think that maybe within the next month or so, we're hoping to migrate this over to CellXGene from the UCSC browser. There's also Genome Browser where you can view tracks, the chromatin tracks, and look at specific regions of the genome, genes that you may be interested in, where you can view accessible chromatin patterns across individual cell types, much like you can again in GEM in the knowledge portal. We also have viewers of links, predicted links between distal sites and genes that Parl's created. Where, for example, gene TMPRSS2, which is implicated in entry of COVID-19, was a gene that we focused on in the eLife paper. You can view all predicted links from TMPRSS2 to other parts of the genome, which is potentially useful in overlaying with genetic data. I think one of the goals for the knowledge portal as well is to incorporate this type of visualization into GEM over the short term to increase the ability to leverage these types of data linking pairs of sites within the knowledge portal. Other tools that we have are to view expression data. So this is something that we set up as part of creating this resource, was a browser of gene expression in individual cells. Cell types, so you can search for genes and it will show you the expression of the gene in multiple ways, the average expression or the percent of cells that were expressing the gene across the different cell types. So again, for TMPRSS2, you can see, preferential expression and alveolar and other epithelial cells. And then you could also break this down by age group or by individual sample. And we're hoping to add additional features as we move forward, additional single cell expression data sets that may have further phenotypic dimensions that we don't have captured here. For example, disease or non-disease. You can view multiple genes at once and so forth. So this is actually fairly handy in order to view the cell type specific expression of genes you may be interested in. Okay. So that's mostly it. A quick overview of the tools in our site, but then of course the key thing is to provide all these data to the Lung Knowledge Portal, and so all the information that I described that we have currently on our site, and as we continue to aggregate more data of this type, passed on to the Knowledge Portal, so like cis regulatory elements, the predicted effects of variants, or actual variant effects, if it's QTL mapping, things like that, genes linked to regulatory sites, the activity of genes, expression, and so forth, and then used in the ways that Maria described in her overview of the site to enhance the ability to understand the function of respiratory risk variants. So thank you very much. So Parl, like I said, did most of the work to set this up, and then also we worked closely with Chin, Sun, Sebastian, and Alan, the Epigenome Center, and then of course all of our colleagues at UCSD and the Broad. So thank you.
That's great, Kyle. Thank you. Questions for Kyle? We have some time. More broadly for other folks.
Like I said, a key thing is in a differentiator of this site from, like, say, the diabetes site, where we have a ton of data, increasingly not even just from diabetes relevant tissues, but just like genomic data generally, is that here this is so far been just focused on the data from this one study, and that we created it for the paper originally and then synced up with the knowledge portal. But we'd like to be more comprehensive of other data types that would make sense to include in our site, that you can visualize there, leverage some of these tools we've created, and also as a means to pass these types of genomic data on to the knowledge portal, which we do quite successfully for diabetes and our other efforts.
That's a really good point, Kyle. Particularly, I think one of the things that we've benefited from in the diabetes community is having like the investigators literally hand in hand with us in terms of what the data sets are. I think the lung community, I see some of you on the call, so I'm happy you're here. And we've just started really to really learn from you in terms of what are the right data sets to go after for both genetic and genomic annotations. I think, having more direct conversations with you about that is valuable to us. So please, if there's things we're missing, and we want to essentially, the way that we started this with Michael and Benji and others is to be sort of the portal and the Epigenome Atlas are really stewards of results representing community-driven, authoritative, if you will, results that become accessible in an integrated manner. We want this to be accessible to useful to all, but driven by experts in the domain. And so we may be experts in software and visualization, but the lung community is really experts in the disease and the biology and the data they're in. So that's the partnership that we're trying to establish with Michael sort of leading the charge on the lung side. But I know there's two, but please, if you are on the call and you are interested in participating, please email me or anyone on the team. We'd love to talk to you more about how to use this, make it better, and how to get the word out and add to it. And hopefully we can look for future updates about merging the two sites and expanding as well. So I thank you, Michael. Thank you, Maria. Thank you, Kyle. This is wonderful. Pardon the technical difficulties at the beginning. You think after a year and a half of doing this, we'd be smooth, but you know, we're all human. Any other questions before we wrap up? All right, again, this will be posted. We'll share the slides. It will be on our website, on the lung site as well, where you can find links to all these things. And stay tuned. We will be back in September for an update on our genomic region minor and the variant exploration tool for prioritizing variants. So take care, everyone. Hopefully, I think the sun's out here finally. So hopefully it is out where you are, and enjoy your day. Take care. Thank you.
Thank you.
Thanks, everyone.
Everyone.